Oh, switching. Thank you.
Everyone, if you don't have an amazing YouTube channel, now you do.
I can see.
Abigail Breland 7776.
Huh?
I will be updating my username to 1776 soon, so I hope you'll still recognize me.
Oh, like 1776?
Yeah, 1776.
You're really patriotic. If you guys, since y'all are doing your warm up, I'm gonna go ahead and stamp the meiosis crash course, which is page 11.5. No, you're fine. That's okay, almost. Okay, guys, let's go ahead and. Okay, so again, we are almost done. I wanted to emphasize that we are almost done. We only have 3 more chapters or 3 more lectures, and after today, we only got 2. I have to say.
I was like, I was just standing by. Like, don't beat me yet.
Do you have your phone?
Yes.
Oh, Jonathan and Liz, your phone's good. Okay, so anyways, so again, we're almost done. Okay, I just want to keep emphasizing that because I need you guys to realize to give me your all because again, we're almost done. Okay, so again, we are covering lecture 11, which is meiosis. Okay, if you would go ahead and read after me or read the bullet parts as we always do. We go number 1,
Discuss
the importance of,
meiosis
and number 2, what
mechanisms,
allow for the increase in.
genetic diversity.
So again, we're going to be looking at meiosis. We're really not going to be focusing on like all of the steps like we did for mitosis. We're really just going to be focusing on what makes it different and like what's the point of these different sections. Okay, that's what we're going to be focusing on. Okay. Alright, so starting us off, let's look at this GIF. Again, we've seen it before. It's the same GIF that we talked about when we looked at mitosis. What are the 3 big differences between mitosis and meiosis? Again, looking at the GIF, what's 3 big differences between mitosis and meiosis? What's one?
Mitosis only divides them once.
So, mitosis only divides once, while meiosis divides what?
Three.
Twice.
Sorry.
But since it divides only once, how many cells does it produce?
2.
While meiosis divides twice and produces 4
4.
cells, so that's 1. What is something different?
The meiosis, each one has 26, so 46, or 23, I mean.
So by the end of mitosis, we end up producing 46 chromosomes, keeping it the same. While meiosis, we end up producing 23. So what do? We halved it. What's another one? Caitlin? Or was that what you were gonna say?
That they're all different and they're not the same cells at the beginning.
So in mitosis, we produce, what's word that means they're the same? There's an I. Identical daughter cells. While in meiosis, by the end, we end up producing all
4 daughters.
4 daughter cells that are all completely
Different.
or unique. Does that make sense? Okay, good. Very, very good. So before we actually start looking, you guys have this image as well, but I want us to review the vocabulary between homologous chromosomes, sister chromatids, and chromosomes. So again, we looked at this in lecture 6, but these are all chromosomes. But more specifically, which one of these 2, 3, 4, or just one, which of these are homologous chromosomes? Remember, there was a word that we could use for homologous chromosomes. What was it? A pair. So which one of these, by the way, you guys did much better on the test, like overall collectively. Very, very happy, okay? Because last time I think y'all were, no, wait, first period was the lowest. They're still the lowest, but you guys did really good, okay? But which one of these is a pair of chromosomes? A, is that the only one?
B.
A and B? Are they a pair?
Oh, no.
No, because they're what?
Connected.
Connected, so they're not a pair. A and who? D as in dog. So A and D are both homologous chromosomes because they are a pair. But what's the difference between A and D?
A has 2 separate ones, and D is the same one but split up.
So not necessarily the same to split up, but these are just a pair, while these pair have already duplicated or have gone through what phase where we duplicate or replicate the chromosomes? S phase. So which one is before S phase and which one is after S phase? D is before S phase while A is after S phase, but they're both homologous chromosomes. This one is before what?
DNA replication.
Or S phase, and this one's after DNA replication or S phase. But if these are not homologous chromosomes, then what are they? Just chromatids or chromosomes. How many chromatids do we have here? Chromatids here? Chromatids here? Chromatids here? 2. 2, 1, 2, 4. Okay. And which of these are, number 2, sister chromatids?
D.
Are they connected? Remember, to be sister chromatids, they have to be connected physically. So this isn't, but A and B. Good. Very, very good. Does that make sense? Again, we're just refreshing our memory because we're going to be talking about homologous chromosomes and sister chromatids. That's what we're going to be talking about. Okay? Again, today we're going to finish everything. Okay? So, let's go ahead and look at this first page of notes. So, let's go ahead and look at the first bullet point. If you would read it for me, please, Nicholas.
Sexually producing organisms are typically diploid or still
Which means that they have a. So, what is 2 and me then? They are who? Anyone. What is 2 and me? They are.
The full set of chromosomes.
What is 2 and me? They are diploid. There we go. Which means they have a full set. But what is 1 N or just N? Haploid. The next chromosome, go ahead, or excuse me, the next bullet point, sorry. Go ahead and read for me, please. Abigail.
These chromosomes are homologous and same shape, size, and genes.
So again, homologous chromosomes, in other words, is just a pair, but that means they have the same size, same shape, and same genes. But more specifically, again, we're kind of, all of exam 4 is what we call genetics. It's all genetics, okay? And typically people do much higher on this test than all of the others, especially with Punnett squares. I don't know what Mr. A does, but you guys can do a Punnett square to like save your life, okay? So, you guys will be fine. But more specifically, what are alleles? Christian, go ahead and read that one for me, please.
Alleles are different versions of a gene. For example, hair color is a gene, but blonde, brown, black, red are all alleles.
So what's another example of allele and gene that we could use?
Color,
Which one?
like eye color,
Eye color. What would be another one?
skin color,
A little bit more complicated, but yeah.
height,
Stuff besides colors.
height,
Huh?
height,
Height. Say what? Face shape. Ears. What about ears? If they are.
attached or unattached,
Huh, where are we getting all these from?
lab,
Exactly. There you go. That was the point, right? Okay, good. By the way, make sure to do your extra credit if you're offered it, okay? Because she did say that she wasn't gonna offer it, but then I guess now because she's not there, they're offering it, so make sure you do it. Because they were going to get rid of extra credit, and extra credit will save your grade.
and
I
your
was-
lecture grade.
I was very, very discouraged. But again, you guys have really, really high lab grades already, majority of you. And with the extra credit would help even more. It literally raises your point or your grade like upwards of 5 points. Please do the extra credit. Have ChatGPT make those questions if you don't wanna write them yourself. Okay, it doesn't really matter. They won't know the difference, okay? It's not like you have to submit them.
But
What?
that's true, you can turn it in on paper,
Exactly,
so
that's what they get.
go.
You never, again, I sit there and look at y'all whenever y'all take y'all's quizzes? That's why they're open up 24 hours. Like, I know y'all are cheating, it is what it is. Like, I'm not worried about it. But anyways, what's my point? Oh yeah, Soraya, read the next one please, one chromosome.
One chromosome is received from the mother and the other chromosome from the father during the process called fertilization.
Again, this is why we see in all of these images, as well as over here, that the red one is representative of your mother and the blue one is representative of your father. But when do those, like it says you get half and half, but they're half, when do they become a whole? During what process? Fertilization. Any time you hear the word ation, it's some type of process, like fertilization, okay? Germination, okay? Some type of process. Matthew, go ahead and read the next bullet point for me, please.
The zygote is the union of both the egg and the sperm cell, and is only one cell, but is a diploid.
So whenever the sperm and egg cells do form a union during fertilization, that one cell is what we call a zygote. And it is diploid because you have half from your mom, half from your dad, you have a whole now. Okay. But the question says, what happens if an organism uses diploid cells for sexual reproduction instead of just haploid cells?
They have double the chromosomes.
It'll double the chromosomes, because, perfect, thank you, like, that saved us like 5 minutes. So we have 2n plus 2n equals what?
4n,
But then 4n and 4n in the next generation would be,
8n,
and then 8n plus 8n in the next generation would be?
16.
So what happens if we don't, use diploid, or haploid cells, we use diploid?
The chromosomes will double.
Chromosomes will double each
Generation.
That's it. Chromosomes will double each generation. I kid you not, Kaylin was just said just like 5 minutes, so.
Thank you for reading.
So what must organisms do though to combat this whole doubling of chromosome at each subsequent generation? We have to
Meiosis.
half it, which is what process?
Meiosis.
Meiosis, that's the whole point, okay? One more time, so what must organisms do to combat this issue? Half the chromosomes, or in other words, meiosis. Again, I want to emphasize, when we're looking at meiosis, we're not looking at the specific steps. We're looking at only what makes them different and what's special about meiosis, like what's the point of meiosis 1 and meiosis 2, okay? Go ahead and read the next bullet point for me, please. Itzel.
Meiosis begins with diploid 2N cells and produces haploid cells.
Savannah, read the next one.
Meiosis is part of the process by which mature reproductive cells called gametes form from immature reproductive cells called
Germ cells. So again, we end up getting who? That are mature sex cells, and what are examples of gametes, which we've already discussed? Sperm and egg. But where do we start off from though? Germ cells, which are immature sex cells. There is no version of it, it's just germ cells, okay? Go ahead and read the next one for me, please, Emily.
Meiosis starts in the same way as mitosis, with DNA replication producing 2 chromosomes with 2 sister chromatids each.
So again, meiosis starts the exact same way that mitosis did, meaning that we end up getting this chromosome right here. These homologous chromosomes, end up going through DNA replication during S phase to produce sister chromatids, but they're still homologous chromosomes. Again, that's what we're getting right here. Almost the exact same thing, okay? The next one, go ahead and read for me, please. Sammy?
The purpose of meiosis 1 is to separate the homologous chromosome to a new daughter cell.
So, looking back real quick, what was the whole point of this whole process to produce what?
Gametes.
Well, are gametes diploid or haploid?
Haploid.
haploid. And what were some examples? They include what? Sperm and egg. By the way, this is going to be our one pager for this chapter. Okay? It's pretty much everything that you need to know is on this chapter. There's a couple things that I left off, but I'll include it tomorrow as well. Okay. And then we start off with what again?
Germ cell.
But what are they? Are they diploid or haploid? What does it say? From immature reproductive cells called germ cells, but meiosis begins with what?
Diploid.
What are those diploid cells called? Germ cells. Good. Okay. And again, what's the point? What's the purpose of meiosis one? To separate what? Homologous chromosomes. Again, let's look. So if we see right here, these are homologous chromosomes. These are what? Homologous chromosomes. But the point of meiosis one is to do what?
Separate them.
Separate these homologous chromosomes so that way that this red one is big chromosome is in this cell, and that large blue one is what? In this cell. Does that make sense? The whole point of meiosis one is to separate homologous chromosomes. To get chromosomes that are from your mom or your dad in one cell, and the other half in what? The other cell. Does that make sense? Okay. Let's move on. What's the point of meiosis 2? Go ahead and read that one for me, please. Abraham.
The purpose of meiosis 2 is to separate the sister chromatids into 4 new daughter cells.
So again, in meiosis 2, we produce 4 cells or 4 daughter cells, but what's the point? To separate what? Sister. Chromatids. Separate sister chromatids. Okay. Go ahead and read the next bullet point for me. Actually, the next 2 bullet points for me, please. Caitlin.
Prophase 1 takes about 90% of the total time of meiosis. Synapsis and crossing over occurs in this step.
So when we talk about meiosis 1, yes, we produce 2 daughter cells, but what's the point of it? To what? Separate the homologous chromosomes. In meiosis 2, yes, we produce 4 daughter cells, but what's the point of it? Is to what? To separate sister chromatids. That's the point. Again, these are homologous chromosomes, but they end up getting separated. These are sister chromatids, but they end up getting what? Separated. Does that make sense? Remember, sister chromatids have to be attached. They have to be attached. Homologous chromosomes are a pair, but again, they're separated in meiosis 1. Does that make sense? Yes? Some of you need to be a little bit more confident with saying, I don't understand. Because you don't come to tutorials, you don't ask for help. You need to be more confident with doing it. Okay? So, one more time. Meiosis 1, what's the point? What are homologous chromosomes? One word. A pair. What's the point of meiosis 2? And sister chromatids are what in one word? Conjoined. Joined or together, whichever word you want to use. They are together, physically connected together. Okay? Do we see that? Right? Okay. And what's the point of, we're going to talk about this in just a second, but crossing over, or what's the point of separating homologous chromosomes and sister chromatids? What's the point? Go ahead and read this bullet point for me, please. No. Nicholas.
To help increase genetic diversity, chromosomes undergo segregation during metaphase 1, 2, and anaphase 1, 2.
What happens during metaphase 1 and 2? They go to the middle. During anaphase, what happens? They get separated towards the to the poles, to opposite cells. Again, that's as far as we need to know. You don't need to know the specifics about the nuclear membrane and all that stuff. It's fine. Okay, that's just it. What does syn mean? Together. So synapsis means something about together, okay? Let's look at these. So in prophase 1, which takes 90% of the total time of meiosis, we look, what kind of chromosomes are those? Okay? Homologous chromosomes, because they are the same size, same shape, and same genes, but more specifically, they have different, there's an A, alleles, okay? So, again, 90% of the total time of meiosis, prophase 1 is occurring, but why? What 2 events are occurring again? Let me look over here. Again, right here. Prophase 1 takes what? 90% of the total time. But why? Because what 2 events are occurring? Looking at the notes right up above. That's the part that Caitlin read. Synapses. And crossing over. We'll talk about this one in just a second, but what is synapsis? Synapsis is this. What do we notice about these 2 homologous chromosomes? The red one from the mom, blue one from the dad. What are they doing? They're like combining together. Not really like fusing all together, but they're just kind of like being glued together. So during synapsis, chromosomes are glued together. Because remember, syn means what? Together. But the second event that occurs is crossing over. But I want you guys to look at the way that these homologous chromosomes look, and then the way that they look afterward. And we look afterward. What do you notice? Again, I'm kind of pointing to it, but what do we see is a little bit weird or different? They take a little small piece off the end. But what piece, what do we mean by small piece? So, yes, a small piece off the chromosome, but more specifically what?
The genetic material.
Close? Alleles. One more time. Why alleles and not just a gene?
Because alleles are specific.
Which could be the same.
No.
They could be the same, or they could be
Different.
They could be different. One more time. What's the point of prophase 1? 2 things happen that take up 90% of the total time. One called what? Which is basically where they're glued together. They form a tetrad because there's 4 parts, 4 little chromatids. But after synapses, or during synapses, we have something called crossing over, where they basically shuffle and exchange alleles. Why? To help increase genetic diversity. So that way all of your eggs or all of your sperm are completely different from one another. Because again, in this one you see 100% from your mom, 100% from your dad. But is it the same here? No. It's a little bit from your mom, a little bit more from your dad, right? More from your dad, or same from your dad and mom, but a little bit more from your mom. This is exactly why sometimes parents or your offspring or your kids in general or your siblings kind of look more like one parent or the other. Because they're getting more DNA from one parent or the other. Okay? But during metaphase, again, they align in the middle, but when they align in the middle, they do so what? When they align in the middle, they do so what? Randomly. What does that mean? That means that not all of your ones from your mom get on the left side and all the ones from your dad get on the right side. They're what? It's everywhere. It's all random. Okay. But during anaphase, when they're separated into these 2 cells, they do so randomly. Again, why are we emphasizing that word random? Because what do we want to do? What's the whole point of producing like a bajillion sperm cells and egg cells that are all different? To increase genetic diversity. To make sure that all your offspring are different so that way they can survive. Okay. Now, in telophase 1, we're only looking at this one cell. Same focus. So, during looking at this one cell, what's question number 1? Go ahead and read it for me, please, Abraham. That's on the back.
And the chromosomes are there, so
So how many chromosomes are there in the cell? Just one. Remember, we count the center. So how many are there? Two. Good. But the second part of the question is what, Abraham? Are they?
homologous chromosomes or sister chromatids.
So are they homologous chromosomes or sister chromatids? They're sister chromatids. Why are they not homologous?
Cause they're conjoined.
Because they're conjoined or joined together or fused together or together. But are they the same size and shape? No. So they can be homologous. Okay. Go ahead and read question number 2, Sammy, please.
How many chromatids are there in the cell?
So how many chromatids are there? 4. Remember, to count chromatids, we count the number of noodles. There we go. Emily, question number 3, please.
How many rounds of DNA replication occurred?
How many times did DNA replication occur? Once. Again, all the way over here, this is before, this is after. Again, looking at here, before all of this happened, we had what? S phase happened. And S phase is where DNA replication occurs. Okay? But it only happens once. Only once. Savannah, read the next question. I think it's question number 4.
Are the cells haploid or diploid?
So are these cells haploid or diploid? Which one? Remember, diploid, you need a full set, or in other words, you need a pair. Do you have a pair? No. So that means they're not diploid, they are haploid. Does that make sense? Why are they haploid and not diploid?
Cause they don't need any other pair.
They don't have the full pair because what did we just do? We just separated.
So for them to be diploid, they have to be homologous chromosomes?
Correct. For them to be diploid, you need homologous chromosomes. Perfect. You said it better myself. Yes.
What do diploid cells look like?
So diploid cells, like right here, they would have the full set of chromosomes, and we're gonna look at it in just a second, but more specifically, they would look something like, not the top one, but the bottom one, where in each cell, you have what? 2 of every single one. One for your mom, one for your dad. One for your mom, one for your dad. Over and over and over. Okay, this is a little different because we'll talk about it in a second. And then after, wait, I forgot about this part. The cell goes through interphase what? After telophase 2. The cell goes through what? After telophase 2, the cell goes through what? Interphase 2, but what does it say? It says interphase 2 is like what? Gap phase 2 or just G2. Do you guys remember what interphase was?
It's when it's just like there.
It's just there, it's just living its life. But what are the 3 parts? Look back over there with that graph. We have G1, S phase, and then G2. But this is saying that interphase 2 is like who though? G2. Not G1, S, and G2. It's saying that it's like what?
G2.
But what does that mean? What's the point of G2 over there? It's making this and this for something. It's making energy, ATP, and making, there's an E. They do stuff. They are made up of proteins. Enzyme. So one more time. During G2, we make ATP and enzymes. To do what? To do mitosis. But during interphase 2, it's like G2 because we're making ATP and, but not for mitosis, but for what? Meiosis 2. Because again, we already replicated the DNA. S phase does not happen because it only happens once. It only happens once. Savannah, how many times does DNA replication occur? Once. How many times does S phase happen, Abigail?
It does not happen.
how many times does it happen overall?
Once?
Once. Because the S phase and DNA replication is
The same.
the same thing. Okay, so I can have a question asking the same thing twice because it's the same thing. Okay. So then after interphase 2, which is like G2, what happens? Now, what does it say for number 5 on the very, very top?
Phase 2.
Then after prophase 2, we got,
Phase 2.
and then, and then, again, notice we're really not even focused on like what exactly happens in terms of the chromosomes, whatever. Because again, what's the whole point of meiosis 2? Over there.
Sister chromatid.
Separate the sister chromatids, that's all that we care about, okay? Again, lecture 6 focused on the other stuff, we're focused on the chromosome. So, same thing, question number 5, go to read for me please, it's on the back.
Oh, right.
Yeah, you're right, okay.
How many chromosomes are in the cell?
So how many chromosomes are in the cell? 2. Are they homologous chromosomes or are they sister chromatids? They're sister chromatids?
No.
No, why?
No, they're not.
They're not joined. They're not conjoined.
But how are they homologous? 'Cause
They're not homologous.
Oh.
Because remember, homologous, same size, same shape. I mean, they're kind of rectangular shape, but they're not the same size, right? Does that make sense?
Yeah.
Again, it is a trick question because we need to understand the whole point was that we already separated the what? In meiosis 1 and in meiosis 2, we just separated the what?
Sister chromatids.
So they're just chromosomes or chromatids. We've already separated them. That's the point. Okay. Question number 6. Go ahead and read for me, please. Matthew?
How many chromatids are there in the cell?
How many?
2.
2. Soraya, number 7.
How many rounds of DNA replication occurred?
How many?
0. Overall is.
Overall.
1.
1. All the way back over here. Okay. That had been a long time ago. But just how many times?
1.
Question number 8, Christian.
Are cells haploid or diploid?
Are they haploid or diploid?
Haploid.
Again, what were they in meiosis 1? Haploid. Can we make diploid cells? No. When do we make diploid cells again? When these two guys in the process called?
Fertilization.
So until fertilization, they're still gonna be haploid, which I just did, okay? Go ahead and read homologous chromosomes for me, please, Abigail.
Homologous chromosomes pair up during prophase 1 of meiosis, and non-sister chromatids exchange segments in a process called crossing over.
So, what do? They exchange segments, but what do we mean by segments? More specifically, they mean alleles, okay? And the second part, crossing over, the second bullet point, Caitlin, go ahead and read that one for me, please.
Crossing over leads to genetic recombination in gametes.
So genetic recombination just means that we're shuffling the deck. What does that mean, shuffling the deck?
So randomized.
Randomize. Again, just like we shuffle the deck of cards, we're shuffling alleles, okay? Yes.
Like, on the right, like, the one that's in the pink square, you see, like, on the one that's blue, it has that tiny bit of, like, pink, is that what the allele is?
Hold on one second.
Okay.
Shuffling the deck of what? Or in other words, alleles. Because exactly that's what we're doing. So if you look right here, the dad has this, what does the capital A represent in terms of alleles? Dominant, while lowercase letters represent recessive. So it's saying that the dad was dominant, the mom was recessive, but they're exchanging, they're shuffling because you're trying to create offspring that are genetically diverse from one another. Because otherwise, if you didn't, then, for example, if you had 2 individuals and they produce like 5 kids, every single kid would be identical to one another, which isn't very beneficial to the environment. Because every single one could potentially have the same genetic disorder or the same problems. But you want kids or offspring that are all different from one another, no matter if you have only 3 kids, 2 kids, or 1,000. We want them all to be different, okay? I don't mean humans, again, we're talking about all organisms, not just humans, okay? Go ahead and fill those in if you haven't already. So, yes, ma'am. Yes, ma'am. Okay, when he's done, or he's not done yet, but when he's done, I'll send him. Okay, thank you, bye. Lance, when you're done, they want you to the office.
Oh, what you do?
I don't think you're going home, they just want.
You're in trouble. Let's go.
Okay, so go ahead and read the first bullet point for me, please, during metaphase. Nicholas, please.
During metaphase 1, homologous chromosomes randomly line up at the spindle equator.
Or spindle, or at the metaphase plate, whichever is fine. Again, the whole point is that when they align it during metaphase, they do so randomly. Abraham, read the next bullet point for me, please.
During metaphase 1, homologous chromosomes separate into 2 haploid cells, each of which with a random mix of maternal and paternal chromosomes.
Again, when they separate going towards opposite poles to 2 new cells or 4 new cells, they always do so randomly. Go ahead and read the next bullet point for me, please, Sam.
Every time a human sperm or egg forms the possible combinations of chromosomes equal 223.
So that means that because of how many chromosomes we have, and from our mom and our dad, when they separate out into a cell, this one sperm cell or egg cell is a 1 out of how many? 223. This one's unique out of how many? 223. Okay. But because each egg cell is 223 variations and each sperm cell is 1 out of 223 variations, when you do the math, technically you as an individual are 1 out of what? 388,608. So when you say like you're 1 in a million, technically you're 1 what?
8 million.
8.38 million.
Is that like how hard it is for people to be twins?
Yes.
So does that mean there could be another male there?
Well, it depends on twins because twins are you talking about fraternal twins or identical twins?
Identical.
But there's identical twins are because there was like during that zygote that forms. What after how do you get more cells?
They divide. They
What is
split
that
from one
called?
egg and go into 2s.
Mitosis, right?
Yeah.
So during the process of mitosis, there's a hiccup, and they instead of like just doing this or like separating outwards, they completely separate. That's identical twins. But fraternal twins are 2 separate eggs were fertilized by 2 separate sperm cells. Hold on one second. So they're completely different. That's why identical twins are always either boys or girls, not boys and girls. But fraternal twins, they can be a boy and a girl, or 2 girls or 2 boys. But technically, there's a 1 in 8 million chance of having fraternal twins that look identical, even though they're 2 separate eggs and 2 separate sperm cells.
How would they
So it
figure
could
that
possibly.
out?
I mean, if they come out, you really couldn't actually,
Yeah.
because they would be almost the exact same. Yeah. Yes, sir. So. She said you have your keys. He said yes. Okay, thank you. Are you almost done? Cause your mom wants your keys. Whatever that means, I don't know. So what's up?
You can give it to her. Is he? Oh well.
I don't
Send
know.
this one.
what's
Oh, you're
your
talking about my question?
Yeah.
What is this? What is this one ask? This question's like very weird.
So it's asking which of these is not a part of those.
Oh,
You have to think of the second picture, not just the first picture. Okay, so now, almost done. The keys?
Yes.
Where are the keys at?
my pocket.
Okay.
I was supposed to be home.
I guess you're not.
Ah.
Okay, go ahead and read the meiosis evolved from mitosis. Emily, go ahead and read the second bullet point, similarities.
Okay, similarities between meiosis and mitosis suggest a shared ancestral history despite the following differences.
Savannah, read the next one.
Similarity.
Or mitosis.
Oh.
Maintains.
Mitosis maintains the chromosome number. Meiosis has the chromosome.
It's all the next one. Mitosis produces.
Mitosis produces genetically identical cells. Meiosis produces genetically different cells.
The next one, Matthew.
Which one?
Mitosis is complete.
Mitosis is complete after one round of DNA replication and cell division. Meiosis also has one round of DNA replication with 2 rounds of cell division.
So again, that whole 3 points of what makes them different is literally just the gist that we talked about at the beginning. It's literally just the gist. Okay. Go ahead and read the next bullet point for me, please. All organisms, sorry.
All organisms have certain genes that code for DNA repair and to regulate the cell cycle. The same genes function during mitosis and meiosis.
So this is just saying, remember that a certain DNA, or certain thing, I can't say it because it's almost the same test, but a certain thing, enzyme, basically proofreads the DNA, right? Again, if you don't know what I'm talking about, then you probably got the question wrong, but there's also a whole bunch of cyclins, which are proteins that govern or check to make sure that the cell's okay during the cell cycle. Okay, so there's a lot of things that are going on. But it's just saying that it's literally the same thing for mitosis and meiosis. Okay. The last bullet point, Christian, go ahead and read for me, please.
Mutations in genes encoding cell cycle checkpoint proteins may have been part of the evolution of sexual reproduction in the processes of meiosis.
Perfect. Moving on. So this last part is just looking at what? Chromosome uh-ohs. So that means there's some kind of error with the chromosomes as we're making copies of them. But these consequences of these uh-ohs can be what? What does that mean?
It won't kill you.
It won't kill the organism, or they can be
Lethal.
which means if they do kill the
Organism.
or mild, what does that mean? Mild.
Not beneficial.
What does that mean in between?
Not as deadly, but it won't really affect you that bad.
So they won't die,
But they'll still do
But
it.
it
but there'll be something weird
A
going on.
little slow.
I didn't say it, Matthew said it. But kind of. That's where we talk about like syndromes, okay? So like Down syndrome is an example of where there's a uh-oh with the chromosomes. Because it is trisomy what?
21.
21. Because if you look right here, looking at these chromosomes, we have an extra what? Chromosome 21. Okay? Does that make sense? But how do these uh-ohs, how are they caused? They're caused by what word? Does it say it there? Can it occur because of non-disjunction. If you would go to read that word for me, please. What is non-disjunction? Abigail?
Is the failure of a chromosome to separate properly.
And when the chromosomes fail to separate properly, they can either affect all of the chromosomes that you have, or they can only affect one specific chromosome. So for example, if you look right here, these are pretty normal, because again, you're supposed to have two, right? One from your mom, one from your dad. But over here, trisomy 21 specifically, there's an extra copy of it. Over here, how many do we have of chromosome number 1? Number 2? Number 3? Again, you're supposed to have what? But it's not even the same between each chromosome.
Oh.
So one more time, we can have chromosome uh-ohs caused by what? Non-destruction, which can either affect every single chromosome, which is called what? Polyploidy, which is this one. Again, every single chromosome is different. You're supposed to only have two. One has two, one has three, one has four, one has five, right? Or it could be aneuploidy, which only affects one specific chromosome. Like this one, which is affecting which one? 21. Does that make sense? Yes.
Was the first picture, is that in a human?
No, that was not human. That one's actually a corn plant.
Oh.
So non-disjunction is failure to what? Separate, which can be polyploidy, which affects what? All chromosomes, or they can be aneuploidy, which only affects what? One chromosome. That's it. And just as a reminder as well, when we gametes are what? One in or haploid. And examples include sperm and what? And when they join, they create what? They create a zygote, and it's one in or two in? Two in. And how do they join up in a process called? Fertilization. But how do we make gametes? We start off with that are what? Diploid, and we can see here that there are homologous chromosomes. But the point of meiosis one is that we produce two cells that are haploid, but why? Because we ended up separating the homologous chromosomes. During meiosis one, which phase lasted 90% of the total time? Why does prophase one last so long? Because they have what? And synapsis is where they are, and what do they form? They form a tetrad. But during crossing over, what do? They exchange alleles, which is called genetic recombination. But the purpose is to increase genetic diversity or variation, okay? But then we end up getting, at the end of meiosis one, we end up getting what? Interphase two, which is like G2, because we're doing what? And for meiosis two. The point of meiosis two is yes to produce 4 haploid cells, but it's also to do what? Separate the sister chromosomes. So we can have 223 possible combinations of one individual egg or one individual sperm. That's the whole point. To make as many baby differences or differences in the babies or the offspring that we produce. Okay. So other syndromes include what? And blah, 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 okay? So, go ahead and look at questions 1 through 14, exclude, or excuse me, 1 through 18, exclude, 14 and 18. I'm gonna go ahead and give you guys about 6 minutes. Your mom has a Camaro?
Mustang.
Oh, Mustang.
I like that.
just put 220. Sure. It'll be okay. About 2.5 more minutes. Okay, so let's go ahead and look at question number 1. So it says, one evolutionary advantage of sexual over asexual reproduction is that it produces what? What do you guys think? More variation among offspring. Perfect. Number 2, meiosis is a necessary part of sexual reproduction because it what?
Reduces the number of chromosomes.
By what? By half. Perfect. Number 3, meiosis, what?
Gives rise to gender diversity more often.
And how does it do it? Crossing over. Synapses is where they're glued together, so it doesn't really aid in that, but crossing over is one. When they align in the middle, do they do so with all your moms on this side, all your dads on this side? They do so randomly. And when they're separated into opposite cells, they do so randomly. Good, bless you. Number 4, sexual reproduction in animals requires what? Do you need gametes? Yes, because gametes are what? Do you need meiosis? Yes, because that's how you make what? Sperm and egg. And you need fertilization because that's how you get a zygote, which develops into a baby, essentially. Because
Is a zygote a baby?
no, it's just one cell. It's literally just one cell. It's not, technically you really don't say baby until after it's born.
Oh, cause it's like a fetus.
Correct, yes. Oh, good point. And then we have number 6. Dogs have a diploid chromosome number of 78. How many chromosomes do their gametes have?
39.
Because it's always half. Good. Number 7. Which of the following cells undergo meiosis?
The diploid body cell.
Germ cells, because what do we start off with? Germ cells, there we go. Number 8, crossing over mixes of what? Alleles, there we go. Oh, and then, number 9, crossing over happens during which phase of meiosis? Prophase 1. Number 10, which phase of meiosis reduces the chromosome number? Anaphase 2. Anaphase 1. Anaphase 1 is when we separate, because that's when we separate the homologous chromosomes. There we go. Okay, so we're gonna go ahead and end there. You guys can do the rest unless you have a specific question. The other ones?
I think you're Richard.
All right, hold on. So let's go ahead and what are these guys? What are these things called?
Posters.
Not just posters, not pamphlets.
Diseases. Mutations.
That's not a disease.
I know, yeah, I know. What's it called?
Local drawings.
Close. They are called infographs.
I said that.
Oh, you did? I didn't hear you. You gotta say it loud. So infographs are what though?
Information on a photograph.
Information, kind of like in a graphic organizer or a graphic piece, but they are not one-pagers. This is something like a one-pager, not a one-pager. And we're not making one-pagers. We're making graphic infographics that express information in a coherent and readable way for the general public. Okay? Again, not everyone's going into science, but this is what we're doing. Okay? So these are a collection of infographs as well as over here that you can see or take inspiration from, but we are essentially going to create genetic disorder infographics with all of this information. So for example, half a point is your what? Which is just the name of your disease. Half a point also is from your name. So if you don't have your name, there's a lot of people that don't write their name on it. Okay? I don't know why. 1.4 QR code to a video or like a TikTok, Instagram, something that either describes the symptoms of that genetic disorder or kind of has, especially like since like on Instagram and TikTok, they have like the day in the life of someone who lives with that kind of genetic disorder, which that's perfectly fine as well. Okay. Who is affected by symptoms, cure, treatment, diagnosis, and creativity. Only this is the 2 points from whether or not if it's an actual infographic or not. Okay, so like this did not get the full. I think it was only like half a point because that's more like a one page. Okay. Again, you can literally finish this tonight, send it to me, done, no problem. Okay. This is not like a whole like 20 week in advance and like a whole poster project or you're gonna record or you're gonna present it or no, you're just making it. I'm gonna put them out like this because I'm gonna print them and you're gonna use them for this last part right here as well as because some of these are also what we're gonna talk about for in the next unit as well, next chapter. Okay. Yes.
Do you want our name on the front or back?
The front.
Okay.
Yes.
And I did. Oh, I thought we can work on this at home.
Yeah.
Oh, okay. No, I was looking at it in Canvas. It said in person, so I was like,
No, it needs to be turned in person.
Oh, okay.
Meaning
like on
that
paper?
you either need to print it off, or if you do send it to me online, then it needs to be in a PDF. Don't
Okay.
share the link with me on Canvas or Canva, and don't try to submit it online because you're not gonna find it. It needs to be submitted either in person, you printed it off on your own, or you send me the PDF so I can print it.
Okay.
Okay, which I'm perfectly fine with doing.
Alrighty.
Okay.
Thank you.
But again, your name needs to be on it, because I do that all the time, where I print it off, and then I just print them, and I'm like, I don't know who this is, and I'm not gonna go through it. Okay, so. These are the disorders, okay? You need to pick one with, 2, then 3 before we pick 4, okay? So for example, no one else can pick, trisomy X, Jacobs, Downs, albinism, or red-green color blindness, or Edwards syndrome, okay?
We can't pick those ones?
No.
Oh.
Not until those are the ones that have been filled up.
Oh.
So that's the benefit of being picked last, because then the other ones might get filled up, so you can pick what you want. Huh?
Could I get number two?
Oh, no. I'm gonna do, okay. First person? Savannah.
Oh.
What you want? If you take too long, I'm moving on. Just tell me the number.
2.
You gotta be another one. We need to fill up 3 and student 2 before we do student 4.
I think we have one option.
Christian?
Oh.
Oh, which one? Which one?
Oh, wait, I didn't get what you meant. So I have to do 12?
No, I'm just saying you can pick anything that's empty in 3 and in 2. And once these fill up, then we can choose 4. Just only choose the ones that are blank in 3 or
Yeah,
2.
so like the one you have, you clicked on,
Yes.
and I can do that?
Mhm.
Oh, yeah, that's what I meant.
Okay, yeah, okay. Christian.
Number 10.
Nicolas.
Number 8.
Itzel.
11?
No. Strike.
12.
That's not right.
Oh, okay, calm down. Trust me, I got my name spelled a bajillion times in my entire life. Isabella, she's got together. Caitlin?
15.
Emily?
9.
Abigail? You can pick Kleinfelter, Hutchinson, Guilford, Progeria, or Tay-Sachs.
I'll do 4. I really want it down to
Abraham? And then Sammy.
16.
Matthew? At the pick of the litter. Which one?
7.
I'm gonna log into your camera. You don't look at
Maybe type down.
Oh.
So,
Yeah.
can I use the restroom?
Yeah. That was on accident. Jonathan, after the test, then you can pick.
Okay.
Okay, so go ahead and write it down so that you know which one you got. You can't access this in module 4, it does say genetic disorder infograph, and there is the link for you to access so you can see it just in case you forget. It's okay.
That's how it. I wonder what mine's about. I never heard of mine.
I think I've done something.
It's XXY.
Intersex! Is that what it is?
Technically, it can be considered intersex, but XXY is where male has an extra X chromosome.
Does that make, if they have that, do they produce more estrogen?
No, it's typically associated with smaller testicles, but the smaller testicles means that you produce less testosterone. Which means, so for example, most men typically, again, a lot of this stuff occurs more often than people think. And so most people that don't have like trisomy X, Klinefelter, or Turner syndrome, most of them don't even realize that until they start trying to have kids as adults. And then you're like, well, why can't I get pregnant? Why can't we get pregnant? Well, it's because you have a low sperm count because you have an extra X chromosome.
I've seen like a video on Instagram and it was a microscope that had little sperms in it, and like they're like not moving. Could that be because of this?
That is true. So sometimes men don't have like motile sperm cells, and if they're non-motile, then they can't swim to the egg, more or less. Or they have non-functional sperm cells, which are just like they're like hopping on one leg and they can't even cross finish.
I feel bad for them. Wait,
They'll be fine.
for this, yeah, they'll die off one way.
Well, there's about approximately 45 million sperm cells per milliliter of semen. They're fine. You can make plenty more.
Yeah. What about if you have 3 X chromosomes? Does that mean you're just like, I don't
know. That's the whole point of this. Finding out.
Oh, I did a presentation on this program as well.
Okay, so again, what I want you guys to go to do, we have 15 minutes left, so you need to go ahead and start making this. Again, you can do this all today.
Turn it into tomorrow.
Oh, when is it due? Sorry.
It's the 11th.
What's the 11th?
Next Tuesday.
And if anyone is out for the stock market challenge, it's still due, doesn't matter. Okay.
Mr. Silva, have you ever like known of anyone who has said the needles when they donate blood are huge? Because I'm thinking about doing it, but I'm scared the needles are gonna be like the size of a pencil.
Yeah.
What?
Yeah. I'm scared, but I want to.
They're not that big.
6 or 7.
Okay. Do they just do it here? Isn't that not a good place to do it?
Why?
Because it gets in your knees.
That's the point.
Oh.
We put the link on the
I'm thinking of an artery.
If-
Arteries are bad. Veins are okay.
And if you haven't donated yet, you gotta.
Yeah, you can have blood, no?
I'm
You can have
nervous.
low.
Mason's like,
I don't know.
You gotta be 180
and
pounds.
who's like,
I'm over that, so I'm chilling.
Guys, again, I'm not gonna have us given any time tomorrow. We have another assignment to complete off lecture 11 before we move on to lecture 12. So we're not gonna have work time. You get this 15 minutes, and that's what we got. Unless you get this stuff done tomorrow, okay?